Hello, everyone, and welcome to today's webinar titled Extracellular Vesicles in Regenerative Medicine. This webinar is part of the Cell Biology virtual event. And I'm Susie Valdez of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots. So let's get started. Before we begin, I want to remind everyone that this event is interactive, and we encourage you to participate by submitting as many questions as you would like during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. If you have any trouble seeing or hearing this presentation, just click on that Ask a Question box located on the far left of your screen. As a reminder, this presentation is educational and offers free continuing education credits. Click on the Continuing Education Credits tab on the top right-hand corner of your presentation window and follow the process to obtain those credits. I now present today's speaker, Dr. Benedetta Busolati, Associate Professor of Nephrology in the Department of Molecular Biotechnology and Health Science University of Torino, and the President of the Italian Society of Extracellular Vesicles. For a complete biography of our speaker, please visit the Biography tab at the top of your screen. Dr. Busolati, thank you for coming. You may now begin your presentation. Welcome.
Hello, everyone. Thank you, Susie, for the nice introduction. As Susie was saying, I am working at the University of Torino in Italy, and that's where I am connected from. So, today's presentation, we mainly focus, as we said, on regenerative medicine, and that's briefly the agenda. I will first go to a presentation on what extracellular vesicles are, showing you how they are important for long-distance communication. I know you had a previous presentation on extracellular vesicles this morning, but for those who connect just now, or for those connecting offline, I will just spend a small time on that. And then I will switch on the focus of this presentation, mainly stem cell EVs for regeneration. And as I am a nephrologist, I will focus on the kidney as a typical organ, but what I'm saying for the kidney is also true for many other organs. And then focus on biodistribution of EVs in vivo, a focus on the cargo of EVs and on the mechanism involved. And finally, I will spend some time on the possible clinical application of EVs, some examples of clinical trials, which are the limits and which are the promises. Okay, so we all know that tissue damage happens, and our organs are in many instances able to put in place some repair. So we have necrosis of the tissues, we have loss of capillaries, infiltration of inflammatory cells, possible modulation of pericytes that transform into fibromyocytes. So what happens? This process of repair not always ends in adaptive repair, meaning we get the organ repaired and the function of the organ. But in some instances, we face a maladaptive repair. So why does it happen? Maladaptive repair may happen due to an excessive damage of the tissue or loss of the cells that could be involved in repairing, could be in charge of repairing the tissue, such as stem cells or progenitor cells. we may lose the vascularization of the organ, so have a vessel rarefaction. Cell senescence, in many instances, compromises the ability of the organ to repair. And it's very important that epigenetic pro-fibrotic changes may happen in those organs. So fibrosis, it's considered a disease of cell reprogramming. That's one example, the pericyte that reprogram into myofibroblasts. So fibrosis is really the example of maladaptive repair, the final ending of the organ function. And also in patients, it's really important that to underline that epigenetic modifications in pro-fibrotic genes are associated with fibrosis development. So why am I focusing on that? That's very relevant for understanding the rationale of using extracellular vesicles and why reprogramming of a whole tissue is really relevant. So, it must be well understood that one single target is not enough to obtain the restoration of organ function. We might target TGF for fibrosis, we might target other genes, but that's not enough, clearly, to get rid of those epigenetic changes. We really need a reprogramming of the whole tissue. And so that's why we are just focusing on extracellular vesicles. Because extracellular vesicles, and I will give you the proof of that, are tiny vesicles that have a very specific and peculiar function in modulating target organ and mediating exchange of their cargo, namely extracellular RNA-se, to reprogram the target tissue. And in that, obviously, stem cell-derived extracellular vesicles have a prominent role. Okay, so what are these extracellular vesicles? That's a definition that I like very much. We need to consider that they are very small vesicles in the range of nanometers, nano-sized vesicles. They are produced by the cell secretory machinery, and they are released in the biological fluid to have an effect on multiple long-distance intercellular communication. So these are electron microscopy images that show you the main pathways of production of extracellular vesicles. So extracellular vesicles is a very general term that comprises different types of vesicles released by cells. So we mainly divide them in small and large vesicles, obviously depending on the size, but also in part related to the mechanism of origin. So vesicles might be released by cells by fusion of multivesicular bodies where these vesicles are present on the surface of the cell. So typically, those vesicles present in the multivesicular bodies are small, and they are called exosomes. Vesicles can also be released by binding on the cell surface. Typically, those vesicles are larger, but that's not always true, and they are mainly called microvesicles. But as a general term, we prefer to call them small and large vesicles. We have, as I was saying, information inside the vesicles, proteins, extracellular RNA, lipids, and all these multitude of factors are actively and selectively compartmentalized within the vesicles. So they resemble the cell of origin, but they are not just the same content of a normal cytoplasmic content of a cell. And in this really fundamental paper, it was shown that if we analyze the microRNA present in vesicles and in cell, they are not absolutely the same. They really separate, and we have small and large extracellular vesicles that even share different types of content. And another important feature of these vesicles is the surface. So the EV surface presents many receptors, many molecules that are necessary for the targeting of target cells of long distance communication. So vesicles can be internalized within vesicles in different ways, endocyted, receptor dependent endocyted, can be fused with the membrane, can be endocyted to plaque dependent endocytosis, and when they release the content, they might have their biological effect. So a large number of studies show evidence that RNA present in vesicles can be translated and can be active. That's just an example using vesicles containing carrier messenger RNA in a reporter mouse in which GFP could be synthesized after getting the carrier messenger RNA EVs. And it's clear that after intracellular, intraventricular injection, we have neurons that are positive for the GFP. And again, also many papers demonstrated the transfer of specific microRNAs to target cells. These are just in vitro experiments show increase in the microRNAs in cells after targeting with extracellular vesicles containing those microRNAs. And on the right, you can also see fluorescently labeled microRNAs that enter into cells, and the target by Western blot was shown to be decreased. And obviously, after 48 hours, the target was re-expressed by the cells. So all this has a relevance in pathology, as it was shown that EVs may be involved in disease propagation. For instance, EVs from mice with hypertension that were administered to healthy mice could transfer the same hypertensive feature. And that's a very from the group of Elliott and Rosenberg. But going to regenerative medicine, the attention goes at the beginning, at least, to EVs from STEM progenitor cells, as they really appear a unique tool in the control of tissue organ regeneration. So the beginning was that EVs may recapitulate the effect of stem cell on target tissues. And they were doing that, displaying long-term tissue reprogramming. And I invite you to go through these two seminal papers that were the first to show the effect of stem cell derived EVs. And mesenchymal stem cells are the best example of stem cells for regenerative medicine. I mean, they are the mostly used in clinical trials, and obviously they have been the first as a possible use of extracellular vesicles in that application. So mesenchymal cells derived from different origins. Bone marrow was mainly the first, and then adipose tissue has a great interest, synovium, umbilical cord, and many others. And they have a combined activity on immune regulation and possible multilineage differentiation. So these cells actively release extracellular vesicles that might be used for regenerative medicine. So these are some electron microscopy studies showing the release vesicles from those cells, and you can very easily see that these vesicles on the right released are very different in size. There are some smaller ones, some larger ones, and also the mechanism is very different of production. Some come from the budding, as we can see in the right panel. Some in the left panel, we can see the arrow showing a multivesicular body that fuses then with the surface. So we must take in consideration that extracellular vesicles released from a cell source in general are a heterogeneous population. And we need to characterize them and possibly to understand the possible function of population of these and their nature and their characteristics, with the idea of going on with possible therapeutic application for those disease. So that's another tool that is very useful in EV characterization, super resolution microscopy. Obviously, working with such small EVs, it's very important that the technology provides tools to analyze, to understand them. And that's happening more and more, with the progression of knowledge. So we can see here vesicles 500 nanometers up to the one on the right is a typical 50, 80 nanometer extracellular vesicle. And there are a lot of criteria that have the International Society of Extracellular Vesicles with the idea of trying to define those vesicles. The expression of markers which are characteristic of extracellular vesicles, we see here CD63, CD9, CD81, and ALEX. Also, we have the NanoView, which is a new instrument that allows us to analyze the surface markers. The MaxPlex, which is a beads-based cytofluorimetric assay that allows the comparison of multiple markers. And so it's very important to show that those vesicles express typical exosomal markers and that they also express markers of the origin, such as mesenchymal stromal cell markers that we can see here, such as CD29, CD146, CD105. And the other side, it's another instrument that allows us to count the number of particles in a supernatant and to analyze their mean size. If we also analyze the content of microRNAs of those vesicles, that's been performed by several groups. This was one of the first analyses, and that showed quite clearly that the processes which are modulated by those microRNAs involved the main pathways of organ development, metabolic processes, compensation. Okay, so then the rationale for the application of those vesicles in models of damage. And I'd like to show you those graphs, which come from a very first paper from our group, in which a mouse was subjected to acute kidney injury, and at day 1, cells, mesenchymal stromal cells and vesicles obtained by the same dose of cells, so the dose equivalent, were injected. And it's very clear, analyzing the function of the kidney, which is here the creatinine and the bone, that a very rapid gain of function was obtained both with vesicles and with cells. And if we also check the morphological appearance, we can see that in mice with AKI, there's a great necrosis. All those pinky elements are cast that represents necrotic tubules with accumulation of proteins. And that's not happening in mice treated with cells, with mesenchymal stromal cells, or with vesicles. But if those vesicles were treated with RNA, it's quite a rudimental but first experiment, the effect of the vesicles was lost. So we also tried to understand where those vesicles were going after in vivo injection, and we can see that in the healthy mice, the vesicles were mainly going into the spleen, into the liver, but in the AKI mouse, the kidneys were a target organ, and you can see also in the lower panels the different kidneys that were recovered 24 hours after extracellular vesicle injection that are all full of extracellular vesicles. That's the red color. So that tells us that after organ damage, the vesicles might accumulate in organs, and that could be possibly due to increasing to damage to the tissue and loss of the barriers that normally compromise this limit, this equation. But we can also speculate that some specific mechanism of targeting might be occurring. And here we can also appreciate how fast is the accumulation of the vesicles in the organ after injection. So we see that after 15 minutes, we don't have much accumulation, but after 5 hours, the organs are already getting a very high concentration. The liver and the spleen are always targeted by vesicles. So those are mainly organs in which the vesicles are cleaved, both in a normal healthy mouse and in the AKI mouse. So we must take that into consideration, and we might also imagine some modification of the vesicles that might prevent the accumulation of them into those organs. And here that's the confirmation just by immunofluorescence of the presence of the vesicles in the, so we can see here in the vessels, after 5 minutes in the upper panel, we have vesicles, and after 15 minutes, the vesicles are already present, tubular structures that appear right here in the right panel. So that's a more recent work showing again a comparison between the cells and the vesicle, which I think is very interesting. Again, the model is in pigs, which receive the diet and then a stenosis of a renal artery to get in 4 weeks a profound damaged kidneys. So we have fibrosis to the kidney, and we can see that both the vesicles and the cells have a very therapeutic effect on limiting the fibrosis of the kidney. But what's very interesting in this paper, the authors could discriminate some peculiar effects of the vesicles toward those of the cells. The cells could preferentially ameliorate vascularization, as we see in the upper panel, the number of vessels with the mesenchymal stromal cells. And at variance, the vesicles show the preferential effect on tissue preservation and the limitation of apoptosis, which is shown in the lower panel on the right. Another example of a long-term administration of extracellular vesicles comes from this paper, in which a diabetic mouse was treated in a curative model after the diabetic onset. The study waited 28 days before administration of extracellular vesicles, once a week for 4 weeks. And again, we can see a very nice effect of the vesicles coming from the mesenchymal stromal cell. I apologize, the slide has some changes. So those were examples. If you go to the literature, you will find a large number of studies showing the effect of EVs in acute kidney injury and chronic kidney disease. Those concepts are solid. The EVs might have a pro-regenerative effect, although there is not a consensus in the site of injection. Different administration were performed, different numbers, even the EV are not counted in the same way. Some studies count them in numbers, some others in microgram, some others in cell equivalents. And that's still a very big limitation in the field to compare different studies. So going a bit more into details in what's happening in the organs treated with vesicles, if we compare by RNA-se, the presence of the heat map of the genes and the control in the disease situation, and in disease situation plus vesicle, we may find that many genes were reverted to the normal conditions. And in the case of the diabetic kidneys, those genes were mainly related to the fibrosis. And it's very interesting to note that a very similar effect was also observed in liver undergoing fibrosis in a NASH model. So that doesn't seem to be typical for the kidney, but rather a general mechanism of reprogramming, as I was telling you. So again, many pro-fibrotic genes were modulated and reverted after treatment. Stem cell-derived extracellular vesicles. So, can we dissect which of the heterogeneous nature is responsible for their biological effect? That's not easy, and several approaches can be taken. One possible approach was to divide the vesicles into small and large by subsequential ultracentrifugation. So we can see here 10 kVs, which are the first going down, which are the large EVs, and 100 kEVs, that are the ones that remain at the pellet after 100 kg centrifugation. So the comparison showed that indeed the 100 K was the main responsible for the renal damage. So the repair of the necrosis and of the casts was gained mainly using the 100 K fraction rather than the 10 K fraction, and that was very similar to what obtained with the total EVs that is in the last column. That normally you understand which are the factors involved in that regeneration if we compare the two populations. That has been performed in that paper, and a number of microRNAs that you can go and check were identified that were mainly involved in proliferative anti-apoptotic pathway. So what's the role of those microRNAs? Are really they the main responsible for the biological release? So some study really suggests that. So microRNA have been cleaved in mesenchymal stem cell EV generating dorsal knockout cells. And then getting EVs from dorsal knockout cells, you might well know that dorsal is involved in the maturation of microRNAs. And then obviously the extracellular vesicles will contain microRNAs. So going directly to the results, we can see that the dorsal knockout EVs were not able to revert necrosis, cast, and renal function. And in the same way, those EVs were not able to reprogram to get that mechanism that we were describing of reprogramming the tissue toward a normal aspect. So that's an analysis of the RNA-se of the tissue, showing that the renal transcriptome of the mice treated with EVs without microRNAs was very similar to that of damaged AKI mice. At variance, the tissue of mice treated with control EVs was comparable, was getting almost a similar transcriptome of that of healthy mice. So is this just what we can speculate? MicroRNAs are the total of the effect of EVs? No. There are for sure also very important other factors, among which we recently studied a protein factor which can be carried by some vesicles present in the urine. So I don't want to make a very long story about that, but just to give you this example, we obtained vesicles from urine of normal subjects, and we found out that those vesicles had an effect that was very similar to that of mesenchymal stem cell derived EVs in terms of restoring the renal function in an acute model of damage. And again, they stimulated tubular cell proliferation, they limited apoptosis. So what was the mechanism? It was very interesting for us to find out that extracellular vesicles from urine at variance of those from mesenchymal stromal cells contain chlorine, which is a factor that is typically released by the kidney and is very important as an anti-aging and regenerative factor. So that's just the proof that the kidney is the principal organ that releases clotting, and mice without nephronic clotting must knock out for clotting, specifically in the kidney, had almost no clotting released in the circulation. So clotting is very interesting, as I was telling you, as it's one of the main factors in our body which controls the aging. Mice without clotting really appear old, and they present the typical features of aging, like endothelial dysfunction, calcification, atherosclerosis, hypertrophy of the cardiovascular system. And clotting is released in the circulation as a small cleaved form, and of interest, the vesicle from the urine carried this soluble small form of clotting. That's another proof that clotting is present on the vesicles. Going on to analyze the vesicles that carry this clotting, not all the vesicles in the urine carry clotting, but rather a large fraction of them. And that's a co-expression of clotting and an extracellular vesicle marker analyzed by super resolution microscopy. So when we administer urinary vesicles, we found out that clotting, which was lost after the damage, was regained in the kidney, treated with urinary vesicles. The same was obtained at messenger RNA level and at protein level. So possibly partly is due to induction of clotting, partly could be also due to transfer. But what's true is that urinary extracellular vesicles and variants of mesenchymal stem cell vesicles had this peculiar effect. And vesicle for mice that were knocked out for clotting, lacked the regenerative effect, showing that clotting was indispensable in the renal protective effect of those vesicles. So that's just an example of a protein factor that could be carried by these and may exert a very important therapeutic effect. And obviously, these factors can be used for engineering inactive vesicles. So we engineered fibroblast EVs with soluble clotting, and again, we got EVs that were active as therapeutic effect. So again, EVs carrying clotting could protect the kidneys after damage at variants of the fibroblast EVs without clotting. Okay, so I cannot go much into details, but I would like to save the last 7-8 minutes for the final discussion on which is the therapeutic potential of those EVs. So we proved mainly preclinical settings that EVs have really a potential interest. They have the possibility to target multiple organs and to reprogram tissue, acting with a synergy of factors, possibly many factors in the content, in the cargo of EVs are doing the job simultaneously, and it's very important that they may reprogram the tissue, they may target at the same time different levels, different mechanisms. They seem to be avoiding some typical stem cell limitation. They are biocompatible They should not have tumorigenic and mal-differentiative effect. And they, as the stem cells, seem to be non-immunogenic. And we had a first feasibility study in which extracellular vesicles from mesenchymal stromal cells have been administered to one patient in a compassionate use. The patient had a very important graft versus host, which was refractory to all therapies and was treated with mesenchymal stromal cells exosomes, showing a great improvement of many of the symptoms of the graft versus host disease. So that's the count of diarrhea. So that's the epithelial damage of the intestine. That's loss of some typical edematotic lesions. That's loss of some other typical necrotic lesions of the graft versus host. And it's not of great difference, the fact that many clinical trials are now starting using EVs for organ regeneration. So the number, the list, if you go on clinicaltrials.gov, is very long. Here are the main that I found just yesterday. Some of them are recruiting, few are completed, and obviously, if we go on the condition, we might say that lung diseases and COVID-19 related diseases are among the list. That's not, that's quite obvious in this period. But we have already results from a first clinical trial, a phase 2-3, that was studying the effect of umbilical cord mesenchymal stem cell derived EVs in patients with chronic kidney disease related to the diabetic condition. So this trial was ended some time ago, was performed in Egypt, and we had, as you can see from the graph, quite a stabilization, if not saying an amelioration of the glomerular filtration rate, which is the kidney function in the patients treated with vesicle in comparison with patients in the control group, which had a decline in the function. Also, the results of the first trial using exosome from bone marrow mesenchymal stem cells are now published, showing safety of the treatment. And again, you can go and check out the paper. What I think is very important in this moment to underline is that there are many open questions and many points that even trials that are done in this extremely specific and peculiar period must follow. And a letter was also sent to the authors of the paper. You can go and read that. So the International Society for Extracellular Vesicles and that for Cell and Gene Therapy underlined the necessity of following several criteria of stringent identity of what is produced and given to the patient. Potency parameters. Obviously, we still need to address many potential side effects. We need very strong criteria for manufacturing EVs, and that must be done in GMP with definition of safety and clinical pharmacology, and we really still need to acquire a lot of experience and knowledge on that, on the conditions for optimal storage, for optimal dosing, and for optimal administration of extracellular vesicles. Also, it's important to make some consideration on the source of vesicles we want to administer. Having potency tests, we need to consider that EVs from different subjects might have a strong variability. So aging and young mesenchymal stromal cells may produce effective or non-effective EVs, has been shown in several preclinical data. And also EVs from diseased patients, such as obese patients, might have strong differences in respect to EVs from cells obtained from normal patients. So there's still a long way to go. I'd like to also show you this very nice slide coming from a web EV talk. In this period, you can find online this AV talk series, and in one of those there was also a presentation by Dr. Fujita of a possible application of autologous non-stem cell EVs, but rather EVs coming from primary cells. So don't forget also the primary cells could also be really important as therapeutic tools. So I just want to end, underline the need of identity, potency, definitions, dose and administration. So we still need to provide many data before we can really go into the clinic with safe products. So thank you all for your attention, and thank you to all the people who contributed to generate this data.
Thank you, Dr. Buzzolati, for that informative presentation. And we will now begin the Q&A portion of the webinar. If you have any questions you want to ask, please do so now. Just click on that Ask a Question box located on the far left of your screen. So let's get started. We have several questions already coming in, Dr. Buzzolati. Why does maladaptive response occur?
So I think, in my presentation, I just pointed out the main possible reasons for that. Aging and exhaustion of mechanism cells that could be involved in repair is one possibility. So we lose with aging stem cells, that's well known. Also the extent of the damage, if we are facing an incredible, very extensive damage, we may not be able to repair it, obviously. But also continuous mechanism of damage, like chronic diseases may lead to important maladaptive mechanism.
And Dr. Buzzolati, why are organs such as the kidney and liver more refractory to stem cell-based regenerative techniques?
Well, I think, stem cell-based regenerative techniques can can be applied to those organs. I mean, in this right moment, we haven't really found an effective therapy. I have to admit that. We don't have therapies that work, I would say, for all the organs. The possible strategies that have been approved by FDA, for instance, that have been really proven to be effective are really, really few. So many organs are refractory, and it's well known that stem cells, when they are administered, they don't really go and transform into the cells of the organ, so they do not regenerate in that sense. They just produce factors such as extracellular vesicles, secretome, many factors that help the organ if it's possible to repair.
Thank you, Dr. Bizzolati, and thank you to our audience members for these great questions. We have time for 2 more questions. What is EV's regenerative success for acute renal failure and for chronic renal failure?
So, as I was saying, we had many data from preclinical experiments, and they show, effect both in acute and in chronic. Obviously, in chronic, a single EV administration mainly fails to repair. We needed multiple administration. I mean, all models have its standard criteria, but possibly for chronic diseases, chronic kidney disease, we need a continuous administration of EVs. But the success in preclinical experiments, even in large animals, has been clearly proven, and we already had 1 trial. So there's some hope, let's say.
Thank you so much. Our final question, are there any environmental or diet factors that promote the life of stem cells in a kidney?
That would be nice to know. I don't think we know and we can answer to that. Diet factor, it's difficult to say. For sure, stem cells in the kidney are not even well described. Possibly there are some progenitor cells, but it's quite elusive. So we know that those cells proliferate. So we need to preserve them, not make them proliferate too much to exhaust their function. But I don't know if there's any factor that helps. That will be interesting to study.
Thank you so much. And actually, we have 1 more question that just came in. Do mesenchymal stem cells seem to be the most promising for kidney regeneration and healing? And if not, which do?
Yeah, those were the first studied and the mostly used in clinical trials. So, they appear the most promising. But I wouldn't really limit to that. I think, we have been studied urinary EVs and many other groups have been studied EVs from renal cells. So it possibly could be that a primary source of cells, which are of vesicles, which come from cells that are of the whole kidney, maintain a signature that helps to restore, to have a therapeutic effect on the organ, rather than simply stem cells, which are very generic, like mesenchymal stem cells.
Dr. Bizzolati, thank you again for your time today and for your important research. Thank you. This was great. Before we go, I want to thank our audience for joining us for today and for their interesting questions, and any questions that we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. And this webcast can be viewed on demand for 6 months until March of 2021. Lab will alert you via email when it's available for replay. And we encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care and stay healthy. Bye-bye, everyone.